Dr. Asha Thomas and Ms. Pranali Jadhav. They will be speaking on the topic computational repurposing of approved drugs using ICM suite, exploring new therapeutic avenues for liver disorders. I am going to introduce them one by one. Dr. Asha Thomas is the professor and head department of pharmaceutical chemistry at Nyan Prasad Global University School of Pharmacy and Research, formerly known as Dr. D.Y. Patil Institute of Pharmaceutical Sciences and Research, Pimpri, Pune. With a career spanning over 26 years, she holds expertise in computer aided drug design, synthesis of new chemical entities, bioanalysis, herbal product development, and herb drug interaction studies. I will now introduce our second speaker of this talk. Ms. Pranali Jadhav is an assistant professor in the Department of Pharmaceutical Chemistry at Nyan Prasad Global University's School of Pharmacy and Research with over 4 years of academic and research experience. She is currently pursuing her PhD under the guidance of Dr. Asha Thomas at SPPU Pune, wherein she is working on the pharmacophore modeling based design of new hepatoprotective agents. Let us all welcome them on stage with a huge round of applause.
Afternoon to all and greetings from DPGU School of Pharmacy and Research. At the onset, we would like to place on record our sincere thanks to Surojit and the entire team at Molsoft and Advent for providing us this opportunity to share our research at this platform. We have heard many sessions across the day, and I think we had a lot of scientific deliberations from great speakers from the industry and academia. We also come from an academic institute, but with computational chemistry and with the use of Molsoft, we are very novice and new users, and we are just going to present some of the research work we carried out using Molsoft in the last few months. So this work is actually the PhD work which Pranali is carrying out under my guidance at the Savitribai Phule Pune University. So moving ahead, we are going to share some of the experiences using the ICM Pro and mainly focusing on exploring new therapeutic avenues for the treatment of liver disorders. You will help me change the slide. So liver disorders, as we all know, continues to pose very significant challenge, affecting millions of people worldwide. If you look at the WHO prevalence rate, they say that more than 2 million deaths annually occur every year because of liver disorders. When we look at chronic liver disorders, the non-alcoholic fatty liver disease is very prominent and very prevalent today in India and has a pooled prevalence of more than 50%. If you look at how liver diseases progress, it is a very rapid progression moving from the inflammatory phase to the fibrosis phase to cirrhosis, then to the end stage liver disorders, and in many times it is also associated with the hepatocellular carcinoma. Now, when we look at liver disorders, some of the major challenges, however, in the treatment, the rapid progression of the disease, or often unnoticed progression to the chronic state. As per the records, less than 2% of people actually affected with the chronic liver disease are actually aware or being diagnosed. Also, there are very limited therapeutic options which are currently available for the treatment of liver disorders. A quick recap into the current therapies which are available. One notable drug is the resmetirom. This drug is the first FDA approved drug for the treatment of MASH and NASH. It is very recently approved. It is mainly a THR beta agonist, and it mainly acts by decreasing the liver fat, inflammation and fibrosis. However, a word of caution, it is associated with hepatotoxicity, causes gallbladder stones, and therefore requires very careful monitoring. Another drug, semaglutide, all of us know, very widely today popular for the treatment of weight control, anti-obesity drug. This drug also has received the FDA approval very recently in August 2025 for the treatment of MASH. It is a GLP-1 agonist, however contraindicated mainly in patients who have a family history of thyroid cancer. Another drug which is being recently explored, tirzepatide. It is a dual GLP-1 GIP agonist and it is showing promising efficacy in clinical trials. When you look at the other therapeutic options which are available today, so they are mainly, we are using anti-diabetic drugs, we are looking at anti-inflammatory drugs, or we are looking at the antioxidant class of drugs for the treatment of liver disorders. Next slide. Now coming to the complex pathogenesis of liver disorders. So if you see, there is an interplay of several pathways, several signaling pathways that are actually involved. Some of the major disposition factors, you have the epigenetic factors which come into play. You have what various environmental factors that come into play, mainly the high fat diet, sedentary lifestyle. You have the alcohol consumption, you have exposure to viral infections. Some metabolic stressors like diabetes, obesity, and also various other altered immune response, and all these interplay of these all events together leads to a cascade of events, and therefore leads to very quick progression to the chronic liver disease state. Some of the core pathways which are involved are enlisted over here, and you can see the interplay of the events in the screen. As we all are aware, natural product inspired drug discovery, a lot of drug molecules which we use today inspired from nature. Even in the class of the hepatoprotectives, we have silymarin. Silymarin, as you all know, very widely used today in India. It is regulated as per the FDA in India and used as a chemical entity for the treatment of hepatic disorders. So for the library, for screening or getting the phytomolecules, we actually went back to marketed formulations today which are available and looked at the phytomolecules which are presented in these herbs. So we initially got as a about 84 phytomolecules, and from these, using various filters, we finally selected a set of 40 compounds with known IC50 value when tested on the Hep G2 cell lines. So if you look at the literature data, all the molecules, we found that they were using different models. Some of them were tested through various other in vitro tests, some of them tested on various other animal models, and therefore to reduce the ambiguity in the data selection, we selected only those phytomolecules which had the IC50 value tested on the Hep G2 cell lines. So these are some of the molecules from the constructed library. Next, we went for the target identification. Here we use the network pharmacology approach. So initially we generated a protein-protein interaction network using the string database. So initially we got a very big network consisting of 97 nodes and 1009 edges. We then applied various constraints and filters. We transported this PPI network to cytoscape, and then we got a very core integrate network wherein 5 nodes and 9 edges were expressed. And some of the major targets that we got through this study were mainly the NF-kappa beta 1, TNF-alpha, AKT1, mTOR, and the interferon signaling pathway. These targets, we then went ahead to look at the gene ontology and the KEGG pathway to understand the cellular locations of these targets, the biological processes they are involved in, and also the signaling pathways involved, especially in case of liver disorders and its related conditions. In the next step, we went for the molecular docking studies. So the 40 molecules which we had already selected, the 5 targets we got from the network pharmacology approach, we went for molecular docking studies. And as I already mentioned, top 5 compounds from each of the docking studies on each of the target, we took it ahead to construct the pharmacophore. So now we were expecting a library of about 25 compounds, 25 leads generated through docking, but we got 14 compounds because multiple of the molecules were acting on multiple of the targets which we had already selected. So we got actually only 14 compounds which had affinity towards multiple targets as implicated here. Next, we went for pharmacophore. So we again use ICM Pro and build the pharmacophore. So we use a common feature pharmacophore using the APF tool, and this is the pharmacophore we generated with the key pharmacophoric features. So as you see over here, a big contribution by aromatic moieties. We have the hydrogen bond acceptors, donors, and even the lipophilic moieties. Against this, we screen the zinc natural database consisting of 1089 compounds, and the top 10 pharmacophore hits we identified based on their predicted pharmacophore scores, which were somewhere in the range of -85 to -88. These molecules, some of the structures are shown over here, and we were very happy to understand that some of the molecules, for example, rosmarinic acid, methyl rosmarinate, the 3-methyl quercetin, when we went back to literature, we understood that these molecules are already studied as good hepatoprotective agents, and therefore we were able to confirm the validity. The next step, we wanted to again validate whether whatever the hits we got, whether they have affinity to these multiple targets that we had already selected. So we did again a redocking experiment on all the 5 targets, and we understood that majority of the molecules, if you see their dock scores on the 5 targets, they are very close to each other, very comparable to the entire library of molecules which we use to build the pharmacophore. From here, we then proceeded for the molecular dynamic simulations. This study we carried out by using the Desmond software. So in the molecular dynamic simulation, we ran a 500 nanosecond simulation run. So the RMSD plot, as you see, low fluctuations indicating stability of the complex. When you look at the RMSF plot, the amino acids which were involved in the interaction with the target, though there is very low flexibility for those amino acids, typically in the range of 1.5 to 2.5 angstrom, and also in the protein ligand interaction histogram, we were able to understand that the amino acids which were involved in the hydrogen bond and the hydrophobic interactions were able to remain in the binding pocket for and have stable interactions with the target. Further on, we will be doing other in vitro studies and in vivo studies for actually establishing and confirming the efficacy of the identified hits. However, till now, whatever work we studied and we did with the ICM Pro, we got good correlations till now with the in vitro predictions and the in silico studies that we obtained. Now for the next case study, I will hand over to Pranali. She will take you through the next case study where we also looked at the drug repurposing approach, wherein we are looking at identifying from among the FDA approved drug database hits which can be taken ahead for their hepatoprotective potential. Over to you, Pranali.
Thank you, ma'am. Thank you so much. Good afternoon, everyone. So our second approach is drug repurposing, which is the finding new uses of already existing approved drugs. So as we know, there are many advantages of repurposing over traditional discovery. From COVID-19, there is large buzz regarding repurposing as many of the molecules was repurposed like remdesivir, favipiravir. There are so many examples. So by checking the data set, we got 12 compounds and we have generated the pharmacophore, which is consisting of lipophilic region and hydrogen bond acceptors, digalol for prostate cancer and ursodeoxycholic acid, which is for primary biliary cholangitis. So here, if we see the identification of digalol as well as ursodeoxycholic acid, which were already reported for the hepatoprotective activities. So it supports our model's predictive power. So we got here confidence that other predicted hits might show activity against the liver disorders. So we have taken that molecules for further assessment. Similarly, so as to validate our pharmacophore, we have performed docking studies. And if we see the docking score of all the compounds are comparable to the co-crystallized ligands which were present on that target. And here for 2 targets, their co-crystallized ligands were not available. So we have used ICM's pocket finder tool for that targets. Then we have confirmed their stability through MD simulation studies and energy calculations by MMGBSA. So there we have found that our complexes were quite stable and they have shown good binding to the targets. Till now we have completed till safety assessment. So now we have performed MTT assay in HepG2 cells. And there our compounds have shown safety profile similar to the silymarin, but yet we have to confirm their efficacy through hepatoprotective assays. So as this study is undergoing, till now our conclusion is that our model is predicting compounds which might show activity similar to the already reported hits. Thank you. Over to ma'am for next.
So to just summarize our experience with ICM Pro, so at the onset, I would like to say we are having a very good learning and enriching experience using ICM Pro. The platform is having a very easy to use GUI interface, a single platform workflow which helps us to carry out a multiple of tasks without dependency on external tools. The workflow integration is very smooth, seamless transfer from one task to the other. And of course, we are also happy with the scoring algorithms because of the good predictive ability of the models that we generated till now. Of course, the study we carried out is limited, but we are very happy that we are able to integrate the Molsoft into our research work that we are able to carry out in our lab. Before we conclude, we come from Dr. D.Y. Patil Institute of Pharmaceutical Sciences and Research. Currently, we have moved to a private university, established our own private university, D.Y. Patil Jnana Prasad University, and we are almost 30 years in the area of pharmaceutical education and research. We have almost all programs we offer in pharmacy, and all our programs were previously affiliated with the Savitribai Phule Pune University. Our institute, for its contributions in the field of pharmaceutical education and research, we have received several other awards. We are NAC accredited among the top 50 colleges in India offering pharmacy education. We also have been listed in the Times Impact ranking. Some of our major collaborators, both national and international collaborators, and some of our funding bodies from which we have received funds through which we are carrying out the research at our institute. So thank you once again, and I would like to place on record my very sincere thanks to Surojit, the entire team at Advent and Molsoft for providing us this platform, and also for all the query solving, all the help that we have been receiving across the past few months, for all the troubleshooting that we have in our lab. And I have already told Surojit we need one more training session immediately in the next month, because we have a lot of queries lined up for you. So thank you once again, and happy learning. We believe that every day is a learning experience. Every day we meet new people, we learn a lot of things from everybody, and we are very happy with the learning experience through the Molsoft software. Thank you once again.
A heartfelt thank you to Dr. Thomas and Pranali Jadhav for the excellent presentation. A round of applause.